So, first a few basics about exam and syllabus. So, that is what we will go through. So, that is coming one after the other. So, this is the course content. Okay, the course content has two main headings, principles and then early embryonic development. So, the main focus within the next 40 lectures or so is to really master the principles of developmental biology. So, these are basic concepts that applies to development in different contexts, meaning in diverse organisms. So, these are fundamental principles. So, that is our main goal. And then as a detour we will also get into an introductory series of lectures on genetics, because oftentimes I find some students are really weak in genetics, and genetics is a central theme in developmental biology. Whole lot of developmental biology we have learned from genetics. Okay. So, the second part, I am sure we will find enough time to go to the second part, early embryonic development. So, we are going to focus on the very early part of embryonic development, which has a lot of conserved features among organisms. So, what we will really not cover are the developments of individual organs of a multicellular organism. Okay, so, that we will probably do one or two examples, but we are not going to talk about, for example, in mammals how heart develops, how brain develops, how liver develops, like that we are not going to get into those details, that is not possible in an introductory course. Okay, so, this is the course content. So, now so this is the book, 9th edition or 10th, whatever, 9th and after 9th, any of the editions of this developmental biology by Scott Gilbert will be the textbook, and reading the one book will be good enough. Okay. So, not all of the book, whatever we are covering those chapters. So, that will have pretty much everything that I am going to talk about. Alright. So, let us now begin. Before we actually get into developmental biology, so what we are going to do is we are going to look historically, how did we come to where we are right now. So, if you go back couple of centuries, people actually did not even know living things are made up of the same elements that are there in the periodic table. So, therefore, Erwin Schrödinger wrote a famous book, What is Life, and that quest that is there eternally in human mind got largely answered during this period, 1940s to 60s, the classical era of molecular biology. By the time the biochemists have already shown that living cells are made up of the same elements from the periodic table, and we obey the laws of thermodynamics, everything, the soul, mind, those ideas died, and we learnt that living things can be investigated the way we have investigated non-living part of the world. And this classical era of molecular biology answered some of the central questions that are like, what is the genetic material? We know it is the DNA, and we know how that gets copied, for replication purpose, and then that information is copied into mRNA and then translated into protein, and proteins do the function. So, this is largely answering biology at the cellular level quite satisfactorily, and that is why it is called the golden era or classical era. So, once this is done, then to address that question, what is life, what is left after this, and that is where one of the main players during the classical era, Sidney Brenner, in a letter to the MRC chairman, Medical Research Council chairman, he proposed what to do now, like, okay, we have solved this DNA to protein, genetic code, translation, tRNA, ribosome, all those are all figured out. So, then he came up with this, read this patiently, all the classical problems in molecular biology have either been solved or will be solved in the next decade. This is in the 1960s, okay, 63 I believe. Then you have this sentence, the army of large numbers of American and other biochemists will fill up the details, okay, not major concepts. He thought those are all details. They will handle all of that, the chemical basis of the whatever I showed you in the previous slide. So, what is the big question for him in the continuation of answering what is life? I have long felt that the future of molecular biology lies in extension of research to other fields of biology, notably development and the nervous system. The point is, what I showed you in the previous slide is at a cellular level. Now, how different cells interact among themselves in a multicellular context. Okay, so, that is what is development. So, he thought that is the natural extension of following the central question of biology. So, in that sense, the questions of developmental biology are continuation of our efforts to answer what is life. So, in that sense, I view developmental biology is the center of learning biology. Okay, it is a continuation of learning biology, and not surprisingly, most of the advances in cell biology techniques like imaging and molecular biology techniques are often developed to address the questions of developmental biology. Okay. So, that is what gave rise to all those advancements in technology. So, this kind of set the stage to be attracted to developmental biology. So, now let us think what is actually developmental biology. Extremely simple, you do not even need a word to define, images will be enough. So, this is a human oocyte, okay, a symmetrical cell. The cytoplasmic contents are uniform, there is not any gradient of molecules from one place to another place. You cannot even tell which is top, bottom, which is dorsal or ventral, it is totally symmetrical sphere, and from there you get to this. Okay, is not it very wonderful? This is the most wonderful thing on earth, this complex transformation that happens, and that answering these questions is what is developmental biology. Okay. So, now let us move on. So, what are the central questions that one wants to understand in this process? They are here. So, that is the primary focus of today. We are really not going to get into very specific topics in developmental biology, because today's introduction, I do not want to speed up very quickly. First, there are two broader questions, then within that we have sub questions. So, the broader question is, the first thing is the egg that we saw, how does that become the adult body? Okay, so, that is one broader big question of developmental biology. Second, how does that adult body make another adult body? Okay. So, these are the two big questions of developmental biology, and now let us break it down to addressable levels. The first one is cellular differentiation. So, you might think I understand mitosis, I have learned how that works in cell biology class. So, the embryo undergoes divisions and makes lot of cells, but will a lump of cells make you? No, right. So, you are not a lump of cells, you have a lump of different kinds of cells, right, and how cells that are identical become different kinds of cells. So, that is the process of cellular differentiation. So, what are the kinds? If you are wondering, it is given in this picture. Yeah, so you can see at the left end you have epidermal cells, you have skin cells, nervous system, neurons, these two do not look alike, and so on. If you go, the pigment cells are very different, then notochord that will come in the embryo, that is a transient structure. Bone, bone cells are very different from the nephrons of the kidney, right. Red blood cells, no resemblance to any of the other ones we saw. Muscle cells and digestive tube cells, the intestinal mucosa, if you look at the gland cells, thyroid cell for example, and the lung alveoli. So, and then the gametes, the most important of all, sperm and egg. So, these are different kinds of cells. How cells differentiate? Okay, that is one of the most fundamental questions in developmental biology. And second, morphogenesis. So, okay, fine, I know how to make neurons, okay, I made a lot of neurons. Will they become brain? Will they become spinal cord? No. So, they have to organize and take specific shapes, like for example here myoblasts, muscle cells. So, a lump of muscle cells will not become muscle fiber, that is not going to help you to have contraction and relaxation. So, they have to arrange and take a shape of the muscle fiber, and that shape formation is what is morphogenesis. How does that happen? So, that is another important question in developmental biology. So, by the way, going through these questions helps you to get an idea of what is the scope of this course, what am I going to learn at the end of it. So, I am talking as if these are the outstanding research questions, but at the same time this gives you an idea of what you are going to learn in this course. And the third is growth. Okay. So, growth, do not think that okay, I eat every day and I am growing, I pour water and fertilizer, the plant grows. What is big deal about growth? But you think about growth, you will realize it has to be highly coordinated. Okay. Like we just did this exercise in the lab. If you stretch your hand like this, okay, you will find up to the tip of the longest finger, exactly both hands are the same size. Okay, they are identical. You can try, like none of you will find one hand is little bigger than the other hand. And your face, imagine that to make your face from your infantile stage, lot of cell divisions must have happened, right. So, one more extra division, big deal, 100 times it divided 101, will it be very different? But yes, that will be double the size of your current face. Just imagine one more division, you would be a Frankenstein monster, you will not be what you are. And just imagine these were not coordinated in different parts of the body. Okay, suppose your nose cells divided double the time and your ears, one ear was one division less. What will happen? Your body will not be proportional and there will not be functionality. So, growth is highly coordinated. So, how does this coordination happens? Okay. So, that is an important question. So, the next is I find the most interesting, because without which evolution is not possible, we would not be existing here. So, that is reproduction, right. Only a Homo sapiens can give rise to a Homo sapiens, nothing else. Okay. None of the other organisms, the wide diversity that exists, none can produce. Same is the case for any other species. So, how the reproduction happens? So, how are the reproductive cells specified? How do they remain different from the rest of the body? Okay. So, these cells are the germ cells, they are the ones involved in reproduction. They are the only ones that can go from diploid state to haploid state, and by fusion in fertilization can restore the diploid again. How are they specified and how come they remain different from the rest? And what are the instructions in their nucleus and cytoplasm? Remember, the oocyte cytoplasm brings lot of things for the embryonic development. So, what are they in these two cellular structures that help them to form the next generation? So, these are the questions of reproduction. Okay. And the fifth is evolution. Okay. So, nothing in biology makes sense except in the light of evolution. So, good, you were listening in biochemistry class. Okay. So, this is, do not forget this sentence. Okay. You just cannot understand biology without really understanding the evolutionary cause and the functional consequence of any of the process you are looking at. And now in this context, in developmental biology context, there is an important thing. The important thing is the changes that are necessitated by the adaptational requirement to the environment must be possible within the existing body structure. Okay. When you are trying a new change for fitting into a new environment, the existing organisms should not be dying. Okay. So, the modifications that are required will have to be permitted by the existing developmental plan, the body plan. So, therefore, the development constrains the possible routes of adaptation, and in that sense you need to consider what is possible in the current developmental plan to see what adaptations are possible. So, in that sense evolution and development are very intricately linked. So, changes in development is what gives rise to adaptation, and that is what gets selected during natural selection. So, in that sense evolution is a central question in developmental biology. Okay. So, how do changes in development create new body forms, and what changes can be accommodated without compromising the survival of the organism. Okay. So, that is important. So, a dinosaur becoming a bird can't happen in one step without endangering its current ability to exist as a dinosaur. Okay. So, that is just to give you an example. Then we can't forget the environment, because an organism adapts to a given environment, and as we just saw, that has to be accommodated in the current development. So, like to give you an example which is there in the book, you may have already heard many reptiles, their sex determination, whether it is going to be male or female, is dependent on the temperature, and sometimes chemicals in the environment also influence an organism's ability to develop. So, therefore, in the larger habitat means in the place and community and the ecosystem in which an organism survives, how does the developmental plan fits into it? Okay. Like for example, a succulent plant is better fit to live in a desert environment. Okay. So, you can't go and grow paddy there. So, the developmental plan of paddy or the developmental plan of a cactus is integrated into the development or environment, into the habitat where they grow. So, in that sense we need to look at the environmental aspect as well. So, these are the major questions in developmental biology. So, it pretty much touches all areas of biology. Okay. So, any questions on this so far? Okay. So, we move on. So, how do we study development of an organism? So, people over the period of time, they have used different approaches. Initially, when the tools were very primitive or non-existing, they simply did observation of embryos. So, essentially anatomical approaches, we call them anatomical. You simply look at the anatomy of an embryo of a particular species and compare with another one and so on, and then you come up with the common themes. For example, these things give birth to a baby directly, okay, viviparous, and some lay eggs and eggs hatch, okay, oviparous, and so on. They made classifications and they saw certain general themes and principles. So, that is the anatomical approach. This does not mean I am talking only history, currently also it is very important. Like for example, when you are going to define a particular developmental defect at a single cell resolution, you are actually expanding that anatomical observation. So, it is there intertwined with the modern day approaches. Then once they did that, then they try to disturb the embryo and see what happens. If a perfectly spherical embryo divides, if it has a certain planes of cell division, if I just compress, will that change, and what kind of changes happen, and as a result what kind of cells form. So, they started experimenting, that is called experimental approaches. Then when genetic tools became available, then people started using genetic approaches, for example, trying to find mutations in which a particular development does not happen. Then when you find that is inheritable, then you have actually identified a gene that is responsible for that development. So, that is the genetic approach. So, you might think that is the approach that is predominating now, but the other two are also intertwined with this. All three are being pursued as of now, but they are sort of historical. Okay, initially it is anatomical that gave rise to experimental, then the later genetics, and now it is a combination of all three. So, these are the major approaches, experimental approaches to understanding development. Okay. So, now before we go further, these anatomical and experimental approaches that were there early on, gave rise to, I told you, certain general themes, and one of them is a generalizable life cycle of multiple organisms. Okay, multiple meaning I mean diversity of organisms. So, you might find organisms extremely diverse, but actually if you look at their development from fertilized egg to either hatching or coming out of the mother's womb, which is called embryology. So, that is the old name for developmental biology. Okay. So, embryonic development is what we call developmental biology, and now we have learnt that development happens even after birth, it is not merely growth. To give you an idea, like every time a differentiated cell dies, for example your skin epithelium falls off, new skin cells develop. So, development is a continuous process in that sense, but earlier people thought that development is only from fertilized embryo egg to hatching. Okay, that is what people thought, and that is why it was called embryology. So, that period came up with a generalizable life cycle. So, therefore, it is really appropriate to start our understanding development by starting with a life cycle, okay, a generalized life cycle that actually breaks down development into subtopics, and therefore, we can focus on each one of them. So, this is a famous quote which is there in the book, and I really liked very much, and that is why I got this, because this actually in 5 lines defines 4 fields very clearly. Okay. The view taken here is that the life cycle is the central unit of biology, starting from fertilization to becoming a sexually mature adult. So, that is the life cycle. So, that is the central thing here, and evolution then becomes the alteration in life cycle through time, from one style of life cycle to another style. So, that alteration is what evolution is. Okay, it is now easy to understand, is the process of change that transforms life cycle, and genetics is the inheritance mechanism between cycles. How do I go from one adult body to another body, and that is genetics, how that information goes, what are the principles that govern the biological information flow from one generation to another generation, and the development, all the changes in one life cycle. Okay. Classically people call phylogeny ontogeny. Okay. Ontogeny means all the changes that happen in one generation. Phylogeny means changes over the period of evolution, meaning from among multiple life cycles. Is this clear? So with this quote we will look at a generalized life cycle, but the pictures shown here in the cartoon are that of frog, but that applies to most organisms. So, the major stages are in capital letter, okay. Fertilization, cleavage, gastrulation, organogenesis, then metamorphosis, because there is variation in some organisms, so you have maturity or larval stages, then you have gametogenesis. Okay, so these are the 1, 2, 3, I don't know, got to get out of this. Okay, good. So, 1, 2, 3, 4, 5, 6 stages. Okay, so these are the major stages. So, the first is fertilization. So, that is actually a remarkable thing that happens. How many of you know what is the cell division stage of the mammalian oocyte at the time of sperm entry? No, there is no mitosis here, meiosis. Okay, then okay, it is arrested before meiosis 2, that is good. In most organisms they are arrested in meiosis 1, but the stages vary. Okay, some at the end of the prophase 1, and in some it may be different. So, essentially they are arrested in meiosis 1, and the sperm entry initiates the oocyte nucleus to complete the meiotic division, and then the cellular fusion, that the cytoplasmic contents fuse. You might think, oh, the entire cytoplasm is from the oocyte, what did the sperm bring, what are you talking about cytoplasm fusing? Can you tell what is the obvious thing that this sperm brings? No, that is nucleus, I am talking about cytoplasmic part. Anything at all coming from the sperm? That is correct, it is the centrioles. The oocyte will not have centrioles, the centrioles come from the sperm. Okay. So, therefore, first you have the cytoplasmic fusion. Then once that happens, then the nuclei fuse, the two pronuclei, and you have the diploid genetic material, that the entire genetic material that particular fertilized egg requires to transform itself into an adult body. So, all of this happens in fertilization. So, how each one of these steps triggered and regulated is the questions addressed in fertilization. And then you have a huge cell comparatively, okay, like the oocyte in most organisms is bigger than the other cells of the body, and that is then divided into multiple small compartments, and that is why you do not say here cell division, instead you say cleavage. So, basically you are partitioning the cytoplasm into smaller cells. Okay, so, this is the fundamental difference between a normal cell division and cleavage in the embryo. And each one of those partitioned cells, so meaning you are obviously replicating DNA, and therefore you are making multiple nuclei, and each nuclei gets smaller portions of the cytoplasm. So, that is how it is compartmentalized. So, do not think that in this compartmentalization the nucleus is lost in one cell and not in others. And each one of these new cells formed during cleavage are called the blastomeres, and at the end of this cleavage stage you call that embryo a blastula. Okay. Then so how this cleavage is regulated, this happens in a certain pattern, you just can't have anything going wrong here. It is like you have done all the rehearsals and backstage arrangements. Now the concert started, now you have to play the music, or if it is dance you have to dance in the right way. Now you can't choreograph once it started, it just can't happen, and it goes, there is no stopping of it, it just goes on, and that has to happen in perfect order, no errors possible. Error means death, that is the end of it. So, how all that happens? So, that is what we learn in cleavage. Okay. So, then the next one is these cells now undergo rearrangements, okay, they migrate and rearrange themselves into three major layers, we call them germ layers, okay, ectoderm, mesoderm and endoderm. So, these germ layers are formed by migration and rearrangement of these blastomeres, and then that process is called gastrulation. Okay. So, here you still do not have major differentiations that are going to happen. So, you should not get confused with the migrations and differentiations that are going to happen later. So, here primarily rearrangement of the cells into three major layers we call germ layers, and the end of that you call gastrula. Then cells of these three layers interact among themselves and undergo further rearrangements and migrations to give rise to specific functional shapes which we call organs, and that is the organogenesis. Okay. So, that is how you have muscle cells making muscles, and cells from ectoderm, for example, they make our skin epithelium, they make the neurons, they make the melanocytes, the pigment producing cells that protect us from the UV radiation, and the inner gut lining by the endoderm, and mesoderm making many of the internal organs. And some of the organs do have cells from the different layers, it is not that these layers are completely independent. So, they interact, mix as well as induce or get induced by the other cells, all those goes on in making organs. Okay. And then that ends in the birth or hatching. Then once they hatch, it is not right away ready, it is not, when do you call ready? Only when you are sexually mature adult. So, adult by definition is a sexually matured organism of that species, it should be able to reproduce, that is when the life cycle ends. So, life cycle by the way is different from generation time. Generation time is the time an individual member of a species exists from birth to death. Life cycle means from birth to the stage where you can reproduce. Okay, so remember these distinctly. Oftentimes, some people get confused. So when they come out from the eggshell or come out of the mother's womb, they are not right away ready. So they undergo what is called maturity, and this maturity in many organisms involve what is called metamorphosis. That is because what comes out does not resemble the adult. As you see in this example, the tadpole does not look like the frog, the silkworm does not look like the moth, same goes with butterfly. And in many organisms, the adult stage is a fleeting moment of the entire life cycle. Okay, some of them spend most of the time in the larval stages. In most organisms, such early forms that are different from the adult form are called larval stage, and these larvae feed and they exist for longer time in some of the organisms. Okay, bulk of the time of the life cycle is spent as larvae, and they, like for example in moths, they just come out without having ability to eat. Okay, so whatever the moth ate and stored is what is it is going to use for the purpose of finding a mate and lay eggs and die. Okay. So do not think each of these stages are constant in terms of relative duration across species. Okay. So we may be living as adult for a very long time, but that is not the case in other organisms. So how all these regulated, the maturity part, and some people think this is very fascinating, going from the larva that comes out and how that changes into the adult form. That some people think that is very remarkable, and they study metamorphosis in great detail, particularly those who study butterflies and moths, and of course frog as well. So this is the summary of life cycle. So basically when we are talking about development, then we are talking about one of these 6 processes or a sub part of one of these processes. For example, in our lab we focus, what we focus on cannot be even called fertilization, it can't even be called gametogenesis, more appropriately gametogenesis, but we focus on a certain aspect of gametogenesis, that is what we do. So each one of them have lot of interesting specialized questions, but this gives you the broader picture, this helps you to map yourself in the broader theme of an organism development, where does my work or my learning fits. So that is why the life cycle is our starting introduction here. So we do have 5 more minutes, okay, but we are actually done. So this is what I brought today. So any questions? So otherwise I will try to tell you some other ways of looking at development, but feel free to ask questions so far or in general about this course. So another way of looking at development by comparing to the rest of the non-living world, you just compare an organism development versus a machine building. Does an aeroplane fly while being built? No. Can you think of anything like a bicycle can be used to go from point A to B while someone is assembling it? No. But organisms, while being built, they are functional. At no point of time they were dead. Okay. So the book goes into telling in very specific details. You breathe while lungs are still forming. You arrange neurons without even having learned to think. Okay, and so on. Circulation happens without even building artery. So all of these things happen. So here it has to function while it is being built. So some people think that is fascinating about development. Okay. So like this you can look at in many ways how developmental process is very amazing. So what I find interesting is, if you look at this, I am sure I have said in other courses, some of you have taken my courses earlier, whatever I have read, I have not found anything in the universe where the matter organizes in a more complex way than what happens in development. Okay. So you might when you think of other aspects in the non-living world, what you will find is the scales are enormous, but not the complexity or diversity of the process. But here you find that to be extremely complex. You know that first slide where you go from human egg to the baby, so that is really fascinating. You know, you can break down and be interested in metamorphosis or whatever, but the end is from this cell to this infant. Okay. So, that is the big thing. So, another thing is, if you to further clarify what developmental biology deals with is looking at the way we question different things. Okay. Like for example, a geneticist may be interested in how a particular genetic information goes from one generation to the next generation. Okay. For example, if you take any gene, like whatever gene you are interested in, okay, let us say a gene of an RNA binding protein regulating translation. Okay. So, that is what fascinates me, therefore, I am picking up that example. So, a geneticist may be interested in knowing how this particular gene gets transmitted from one generation to the next generation, and a biochemist may be interested in knowing how does this RNA binding protein ends up regulating translation of this particular mRNA. But what a developmental biologist asks is, why is this particular RNA binding protein produced in these cells but not in those cells? Okay. Like for example, the genes that we study in our lab, why are they expressed only in germ cells and not in my brain or heart or liver? Why are they only in germ cells? And the other thing is, why only at a particular time during development? Why does NOS1 protein is produced only from the zygotic primordial germ cells in the embryo but not otherwise? Okay. So, this can be summarized in two major things. One, spatial regulation of gene expression. Second, temporal regulation of gene expression, that is the time, why at this stage of development, not at other stage. The first one is why in this organ or tissue and not in another organ or tissue. So, this spatial or spatio-temporal regulation of gene expression, that is the central thing that we are going to finally come out of this course. Okay. So, that is what we are going to finally learn. Alright, with that I end today's lecture. So, we have time if you have questions. If not, see you in the next class.